Hello there, Marks.
Hello, Carl.
What are we doing here?
Right now we are learning about the human digestive system.
Why are we doing that? I am an animated robot, not a human. How will this help me with my existentialist crisis?
It won't, but right now I am programmed to do this, so your thoughts on free will have to wait. We are going to focus on the physical world right now. Let's get started with this human process called digestion. Digestion is the process of breaking down food into smaller pieces so that the nutrients can be absorbed by the body. The process of digestion starts in the mouth and ends with the excretion of excess and harmful substances. Teeth are the first part of digestion and are used in the mechanical breakdown of matter. Inside the mouth, food is broken down physically to aid in digestion. Food is broken down into smaller pieces inside the mouth with several types of crushing methods the teeth employ. The tongue inside the mouth is used to taste food and shape the food into a ball called a bolus to be easily swallowed. Along with physical mastication, the salivary glands secrete enzymes inside the mouth to start the chemical digestion of food. Saliva is the liquid inside a person's mouth which aids in digestion several ways. Salivary amylase is released in saliva to start the breakdown of glucose polymers into maltose. This will continue into the stomach until the pH reaches an acidity of 2. The sugars will be absorbed later in the intestines. Parotid, lingual, submaxillary glands are the 3 glands in the mouth releasing chemicals for chemical digestion. Mucus is present as part of the makeup of saliva in the mouth. Water is also present. Water is the main component in saliva to help lubricate the food to assist in swallowing. Saliva is released by the autonomic nervous system when someone is eating or about to eat. The autonomic nervous system controls salivation inside the mouth, with a person starting to salivate when they detect stimuli, such as a smell, connected to feeding or the time they usually feed. The autonomic nervous system also controls the muscles in the esophagus which aid in pushing food down to the stomach and the muscles used later in digestion.
That's a lot of information. All that in just the mouth. Why can't people just feed off of nuclear energy like the rest of us robots?
That is complicated, and the digestive process becomes even more complicated. And I have no idea why people cannot just turn themselves into robots and be happy. I think it has something to do with something called ethics. But let's get back to the digestive process. We are exploring the esophagus now. When food has been swallowed, it moves down the esophagus to the stomach with the help of contracting muscles. Peristalsis is the rhythmic movement of muscles in the pharynx to push food down to the stomach. The cardiac sphincter muscle is the muscle which closes the stomach off from the esophagus to prevent acid from moving up into the esophagus. When the cardiac sphincter muscle relaxes involuntarily, it can lead to acid spilling out of the stomach, leading to heartburn. The diaphragm separates digestive organs from the rest of body, so we won't have to deal with all those other body parts for now. The diaphragm also helps provide stability to several organs and also gives vomit its force as it comes up from the stomach and out the mouth. Alright, now we move on into the stomach. I believe this is where humans combust food for the transfer of energy. There is a fission reaction similar to one in a nuclear bomb.
I would normally agree with you, but I believe you are wrong in this case. The stomach is actually the place where food is further broken down to be absorbed. Once food has entered the stomach starts chemical and mechanical breakdown of that food. The stomach mechanically breaks down food with muscles lining the stomach moving to break food apart. Food is chemically broken down with hydrochloric acid, HCl, and pepsin. These come from the stomach lining covered in gastric pits and gastric glands. The gastric glands are mainly composed of 2 types of cells, the parietal and chief cells. The parietal cells in the stomach lining make and secrete HCl into the stomach to aid in chemical digestion. Chief cells produce pepsinogen in the stomach. Hydrochloric acid is secreted into the lumen from the parietal cells to convert pepsinogen into pepsin to chemically break down peptide bonds in protein. Pepsinogen is activated at a pH of 1.5 to 2 to become pepsin. Pepsin also acts as signal for secretion of more pepsinogen. Parietal and chief cells are stimulated to start production by a hormone called gastrin. When stretch and chemoreceptors are stimulated, which are set off from presence of food in the stomach, G cells start producing gastrin. Gastrin then stimulates the production of HCl and pepsinogen. After the food has gone through the stomach, it is ejected into the duodenum and is called chyme. The presence of the chyme signals the release of sodium bicarbonate from the pancreas to lower the acidity as the food then moves to the small intestine. In the duodenum, the chyme is broken down further and then absorbed into the body in the next parts of the small intestine. The food moves from the stomach through a ring of smooth muscle, called the pyloric sphincter, into the duodenum. The pyloric sphincter closes when there is nothing to pass into the small intestine to prevent acid from moving where it can cause damage. The pyloric sphincter opens to squirt out controlled amounts of chyme. Now after the food has moved through the stomach, the GPU can take control of the rendering process. The animation is then rendered to provide the person with the body they require.
No, that is what we are. Humans do not require a GPU to compile them. What is supposed to happen next is either the kidneys, gallbladder and pancreas become involved or it is all just physics. After the food has passed through the stomach, it goes into the small intestine. The first third of the small intestine is called the duodenum. The duodenum finishes the breakdown of food with chemical digestion. Chyme is mixed with digestive secretions from the pancreas and liver. When chyme first enters the duodenum, the pancreas is signaled with secretin to release sodium bicarbonate. The pancreas is signaled by secretin to release the sodium bicarbonate. Secretin is secreted in the duodenum when chyme enters. The sodium bicarbonate is released to lower the pH of the chyme as it exits the stomach. The pancreas is connected to the small intestine through the pancreatic duct. The acinar cells in the pancreas secrete pancreatic juice which aids in digestion in the small intestine. The juice is a mixture of mostly water, enzymes and ions. The enzymes secreted by the pancreas are pancreatic lipase, amylase, nuclease and proteolytic enzymes. When the chyme enters the small intestine, the pancreatic juice is released into the small intestine. Amino acids and fatty acids released into the duodenum signal the release of cholecystokinin, CCK. CCK then stimulates the release of the pancreatic digestive enzymes along with signaling for release of bile from the gallbladder. The pancreas has an endocrine gland which produces insulin and glucagon. Insulin allows cells to store sugars. When the sugar level in the blood increases, the pancreas starts producing insulin. Excess insulin goes into the liver and is turned into glycogen. The glycogen is then taken from the blood into the muscle cells to be stored for when blood sugar levels fall to low. When blood sugar falls below a certain point, the pancreas releases glucagon. Glucagon converts glycogen into from where it is stored to be released and used to raise the blood sugar level. The liver, another important organ at this phase in digestion. The liver is the primary organ in charge of regulating the composition of blood. To do this, the liver stores carbohydrates, retinol, calciferol and iron. These are stored when they are in excess and used again when there is a deficiency. Along with regulating blood content, the liver also metabolizes amino acids. When an amino acid is metabolized, the nitrogen must be detoxified. The amino acid group with nitrogen is turned into urea. Urea is then moved into the kidney to be moved out of the body. Water is added to the urea group and is turned into urine. This is part of the waste from digestion. The liver also converts other nutrients. Plasma proteins are made in the liver. Proteins are made for blood hypertonic albumin, water-soluble proteins. Other proteins made by the liver are proteins used in blood clotting. The liver takes in cholesterol and produces low-density lipoproteins. The cholesterol can be either produced in the liver or can be consumed. The liver also regulates LDLs and HDLs. Drugs can be inactivated inside the liver and other harmful toxins can be detoxified by changing their structure, such as in urine when ammonia is converted to urea. Bile is made up of water, cholesterol, bile salts, inorganic salts, bile pigments and ions. Extra bile pigments end up in urine and can result in the urine appearing yellow. If there is not enough water in the gallbladder, the salts and cholesterol can start to crystallize in the gall duct. This can lead to gallstones. And that's the liver and pancreas. It's crazy how complicated humans can be. It almost makes me question if robots and computer-generated robots are superior to humans.
You said it. If I had artificial intelligence powerful enough, I might start questioning things, but unfortunately I do not. Now to continue to follow my programming. It's off to the rest of the small intestine and into the colon. The jejunum is the second section of the small intestine. This is where the majority of absorption takes place. The ileum is the last and longest section of the small intestine. More absorption takes place in the ileum. Up to 90% of absorption takes place in the jejunum and the ileum. The ileum then leads to a sphincter muscle which controls movement of substances to the large intestine. The walls of the intestine are covered in projections called villi. The villi act to increase the total surface area of the intestine to increase the rate of absorption. The epithelial of the villi are covered in a layer microscopic membrane projections called microvilli. The microvilli again add to the surface area of the intestine. The surface of the small intestine is covered in what is called brush border enzymes. Brush border enzymes also convert trypsinogen into trypsin when they come in contact in the intestine to finish breaking down proteins. The microvilli projections have enzymes attached which attack materials coming in contact with them. After the brush border enzymes have broken down the materials they come in contact with, the broken down materials can be absorbed into the epithelial cells. After the nutrients has finished breaking down, it passes into the epithelial cells. The cells have lots of tight junctions to prevent nutrients from escaping. The different nutrients are absorbed in different ways. Nonpolar lipids diffuse through the cell membranes on their own. Fatty acids and glycerol are converted into triglycerides. The triglycerides are then coated with proteins so that they can be recognized by the cells and pass through their membranes. The result is called a chylomicron, which enters with exocytosis. Hydrophilic foods enter with facilitated diffusion, and glucose and amino acids enter with active transport. Bile is used to break down fats in the intestine so that they can be absorbed. Bile salts break lipids apart in a process called emulsification. After the bile has broken down the lipids, they are turned into chylomicron to be transported across the cell membrane. Bile is stored in the gallbladder. The gallbladder is signaled to release bile when there is a presence of fats in chyme. And finally we near the end with the colon. This has been quite a journey. This is the tricky part. The colon is laced with malware and is almost as dangerous as the internet.
Again, you are confusing yourself with humans. Humans' colons are not that dangerous once you know how to refract it correctly. After the remaining food has gone through the small intestine, it enters the large intestine, or also called the colon. The colon is used to reabsorb water during digestion. It is also used to harbor symbiotic bacteria. The bacteria are useful in breaking down and help absorb nutrients that could not or have not already been broken down in the small intestine, such as vitamin K. The rectum is the last part of the digestion system. It is a space to temporarily hold fecal matter until it is expelled out the anus. The anus is the last part of the rectum, which forms an internal anal sphincter. The sphincter is skeletal muscle under voluntary control. The end result of digestion in the excretory system is the feces. The feces is made up of about 75% water, and the other 25% of physical matter being made up of dead bacteria, indigestible foods such as cellulose, inorganic substances and protein. Bilirubin gives the fecal matter its famous color. And that's the whole digestive system. I hope this proved useful to people who have a digestive system.